মিউট আছেন, কাইন্ডলি স্যার আনমিউট করে নিন প্লিজ।
শুধু দা মিউট আছেন। হ্যাঁ শোনা যাচ্ছে, বলুন বলুন, শোনা যাচ্ছে।
প্রথম থেকেই বলবো নাকি?
হ্যাঁ বলুন।
Honorable President Sir, who is also my teacher and inaugurator of this program, Professor Dr. Subrat Roy, Executive Committee members of the Zoological Association of Ward 1, the resource persons, the students, our association, the Zoological Association of Ward 1. The participants know that from its very inception, it always acts to promote the advancement of learning in zoology. In our association building, the Professor Dev Kumar Chaudhary Bhavan, we have been running a library, the Professor J.N. Mehta Library, since the last 21 years, mainly for the benefit of the honors and postgraduate students of zoology. In recent past, we donated teaching aids to several institutions. All we know that due to COVID-19, the teaching learning process in all the spheres, that is secondary, senior secondary, and higher education, that is primary, secondary, senior secondary, and higher education, we have been severely affected. Students have not been able to gather information in their respective field of studies. Now, the institutions have been reopened 16th of this month, that is last 16th of this month, but full-fledged operation of offline method is yet to start. On the other hand, the third wave of COVID showing its red eyes. We don't know how many days the educational institutions will remain open. In this situation, we are very eager to do something for the benefit of the students. Accordingly, the executive committee promptly accepted the proposal of some honorable executive committee members, especially Dr. Manabesh Majumdar, Dr. Saroj Kumar Ghosh, and Dr. Samadas Bandopadhyay to organize online lecture series for undergraduate students in undergraduate studies in zoology. In this way, we are trying to give quality online lecture for the students of zoology. Now, in this program, our Honorable President Sir, Executive Committee members, including office bearers, resource persons, and student participants. At the end, I am to say that we will be very happy the students be benefited. I wish the program a grand success. Thank you all.
ডক্টর মন্ডলকে তার মূল্যবান বক্তব্যের জন্য অসংখ্য ধন্যবাদ ও কৃতজ্ঞতা জানাই। এখন আমি আমাদের অ্যাসোসিয়েশনের সভাপতি ও বর্ধমান বিশ্ববিদ্যালয়ের প্রাণীবিদ্যা বিভাগের বরিষ্ঠ অধ্যাপক, আমাদের সর্বজন শ্রদ্ধেয় ডক্টর সুব্রত রায় মহাশয়কে উদ্বোধনী বক্তৃতার জন্য সনির্বন্ধ আহ্বান জানাচ্ছি।
Thank you, Manabesh. At the very outset, I offer sincere thanks to all students of different colleges who have shown interest to participate in this lecture series. Thanks are due to the academic community of those colleges who have motivated the students to take part in this lecture series. Well, it is most likely that since the deliberation are in English, the inaugural address should be made in English, and I was ready for that, but some of my beloved students, with special reference to Dr. Manabesh Majumdar, who happens to be one of the organizers and who happens to be a senior faculty member of the Department of Zoology, Vijayanarayan Mahavidyalaya, requested me to deliver my inaugural address in Bengali. he told me that he had the opportunity to listen to so many deliberations from me, and he will be rather happy if I try to deliver my lecture in Bengali. বন্ধুগণ, আমি সবাইকে আন্তরিক ধন্যবাদ আমার বক্তব্য শুরু করছি বাংলাতে। আশা করি আপনারা সেটা মেনে নেবেন। এখন প্রথম কথা হচ্ছে কি, ছাত্র বন্ধুরা সবাই জানে যে গত 2 বছর ধরে কোভিডের মারাত্মক আক্রমণে সমস্ত মানব সভ্যতা বলতে গেলে প্রায় বিপর্যস্ত। সুতরাং এবং সেই প্রেক্ষাপটে সমস্ত মানুষের সমস্ত অ্যাক্টিভিটি বা তাদের কাজকর্ম ব্যাহত হয়েছে এবং তার থেকে বাদ যায়নি আমাদের শিক্ষা জগৎ। কারণ বন্ধুরা সবাই জানেন যে এই কোভিড আক্রমণে এবং অনন্ত দীর্ঘায়িত লকডাউনের প্রভাবে আমাদের সমস্ত একাডেমিক প্রোগ্রাম ভীষণভাবে ক্ষতিগ্রস্ত হয়েছে, ছাত্ররাও ক্ষতিগ্রস্ত হয়েছে। এইসব কথা চিন্তা করে আমাদের জুলজিক্যাল অ্যাসোসিয়েশন, যার সম্বন্ধে ডক্টর সুব্রত মণ্ডল একটু আগে কিছু বললেন, তারা একটা বলিষ্ঠ পদক্ষেপ নেওয়ার চেষ্টা করেছে যে কলেজ ছাত্রছাত্রীদের জন্য একটা লেকচার সিরিজের ব্যবস্থা করা বা আয়োজন করা, এবং সেখানে যদি তারা অংশগ্রহণ করে নিঃসন্দেহে তারা বিশেষভাবে উপকৃত হবেন। আমার মনে হয় যে সমস্ত কলেজের যারা অধ্যাপক তারা আন্তরিকভাবে আগে চেষ্টা করেছেন যে অনলাইন লেকচারের মাধ্যমে ছাত্রছাত্রীদের যাতে এই সমস্ত যে সমস্ত ক্লাস বাদ যাচ্ছে সেগুলো পরিপূরণ করে দেওয়ার, তার টপিকগুলো। এই কথা অনেক অনলাইন লেকচার করেছেন, কিন্তু দুর্ভাগ্যের বিষয় সব ক্ষেত্রে কিন্তু সফল হওয়া যায়নি। কারণ যদি আমরা সার্বিকভাবে চিন্তা করি তাহলে দেখব যে অনেক ক্ষেত্রে বিশেষ অসুবিধার সম্মুখীন হতে হয়েছে, তার ফলে অনেক কিছু বিষয় তাদের গোচরে আনা যায়নি। তো এই কথা ভেবে যে আমাদের জুলজিক্যাল অ্যাসোসিয়েশন বর্ধমান বিশ্ববিদ্যালয়ের স্নাতক পর্যায়ের যে সিলেবাস আছে তাকে কেন্দ্র করে কতগুলি লেকচারের বন্দোবস্ত করেছেন, যার মাধ্যমে তারা ছাত্রদের ওয়াকিবহাল করতে পারে। এবং আমার বিশ্বাস যে সমস্ত ছাত্রছাত্রী এই প্রোগ্রামে বা অংশগ্রহণ করবে তারা নিঃসন্দেহ উপকৃত হবেন। কারণ এই সমস্ত লেকচার সিরিজে যারা তাদের বক্তব্য রাখবেন বা ছেলেদের কাছে প্রাণীবিজ্ঞানের বিভিন্ন ধারা তুলে ধরবার চেষ্টা করবেন, তারা হচ্ছেন বিশিষ্ট বিভিন্ন কলেজের বরিষ্ঠ অধ্যাপক, এমনকি অনেক বাইরেও যারা গবেষণায় বা অধ্যাপনায় নিযুক্ত আছেন, ভারতের বাইরেও, তারাও কিন্তু এই অনলাইন প্রোগ্রামে অংশগ্রহণ করছে। সুতরাং আমার বিশ্বাস ছাত্ররা এতদিন ধরে যে শিক্ষায় যে চলছিল, বাধা বিপত্তির সম্মুখীন হচ্ছিল বারবার, এবং তাদের পড়াশোনার খুবই ক্ষতি হচ্ছিল, সেটা কিছুটা পূরণ করতে পারবে। আমি অধ্যাপক যারা এই প্রোগ্রামের অর্গানাইজার, যাদের প্রচেষ্টা এই লেকচার সিরিজ সম্ভব হতে চলেছে, তাদের কাছ থেকে আমি জানতে পেরেছি যেটা এটা বেশ কিছুদিন ধরে চলবে এবং যত সংখ্যায় ছাত্রছাত্রীরা যোগ দিতে পারে সেদিকে তারা নজর রাখবে। আমি আশা করি যে এই সমস্ত লেকচার সিরিজে অংশগ্রহণ করার পর ছাত্রছাত্রীরা তাদের যে ঘাটতিটা হয়েছে বা আছে সেটাকে পুষিয়ে নিতে পারবে। আমি এই যে এইভাবে এই ইনঅগারাল অ্যাড্রেসে এই কটা বক্তব্য রেখে এবং ইনঅগারাল অ্যাড্রেসে এবং এই প্রচেষ্টার যাতে সর্বাঙ্গীন ভাবে সুন্দর ও সফল হয় সেটা জানিয়ে আমি আমার ছোট্ট বক্তব্য শেষ করছি। নমস্কার।
স্যারকে তার মূল্যবান বক্তব্যের জন্য অসংখ্য ধন্যবাদ এবং কৃতজ্ঞতা জানাই। এখন আমরা আজকের নির্দিষ্ট বিষয়ের বক্তা ডক্টর সৃজিত খান মহাশয়কে উপস্থাপিত করতে চলেছি। আজকে বক্তাকে উপস্থাপিত করার জন্য আমি বর্ধমান রাজ কলেজের প্রাণীবিদ্যা বিভাগের সহকারী অধ্যাপক ডক্টর শ্যামদাস বন্দ্যোপাধ্যায় মহাশয়কে আজকের বক্তাকে উপস্থাপিত করার জন্য আমন্ত্রণ জানাচ্ছি।
Thank you very much. Good evening, ladies and gentlemen. It's indeed a pleasure to have you all around for this interactive session this evening. being organized by the Zoological Association of Burdwan. To begin with, we have Dr. Srijit Khan, an immunologist by profession, currently associated with Amaris Consulting, part of Sanofi Pasteur Limited, Canada. Also happens to be and a former member of this esteemed association. Dr. Khan, born and bred in this town, who pursued his bachelor degree in zoology from Vivekananda Mahavidyalaya, Burdwan, follow on masters in zoology from Banaras Hindu University, and then on he completed his doctoral degree from the National Center for Cell Science, Pune. Later, he decided to pursue his career in the Royal Nation of Canada. During his earlier days, he was a former senior scientist in GE Healthcare, also postdoctoral fellow in University of Toronto. Feather on the cap, Dr. Khan has authored and co-authored publications in peer-reviewed journals, which are of international standards. Let's give him a big hand and begin with the session. Please, Dr. Khan.
Thank you, Shyam. I would first like to thank the organizing committee of Zoological Association of Burdwan, especially Dr. Rai, Dr. Mujumdar, Dr. Mondal, and Dr. Bandyopadhyay, and also my respect to Dr. Guhu, who has been my teacher at Vivekananda Mahavidyalaya. So currently, I'm working as a scientist in a company which produces vaccines, pediatric vaccines, and I had my training in my zoology in Burdwan in Vivekananda Mahavidyalaya, which started my interest in biochemistry and molecular biology. Moving on to my master's degree in Benares, which also solidified my career course, and later moving into research. I chose the path of working on immunology, which I've carried forward. So I think where I am currently in my career is all, due to the blessings of my professors who are there in the university, and all, my peers from whom I have learned a lot. And it's a great opportunity for me to give back something. And I hope that the lecture that I'm going to give on split genes today is going to help the students. So dear students, I'll start the lecture, and I hope that this will be useful for the completion of your course, and you get motivated to learn more on molecular biology, and it helps you choose your career, or it grows your interest in zoology and the branches of zoology in general. So I'll share my screen. Please let me know if you are able to see the screen in full screen.
Yes, we can.
Yeah.
Please go ahead.
Okay, so the topic that I was that I had initially decided that I would give a talk on was the split genes. I mean, if you see the entire syllabus in the syllabus, the part of Unit 5, which is the post-translational modification and processing of eukaryotic RNA, it's a pretty big part of the syllabus. You have 8 classes dedicated to it. But so I've tried to concentrate only on a certain part, because trying to teach everything in one class would be too much information at one go. So I'll just talk on the split gene, the concept of introns and exons, the splicing mechanism, and focus on a part of alternative splicing. So just to get a refresher of the basic concepts, this is something that we all know. Just to refresh your mind, the organism is an individual form of life. We know it must show growth and reproduction. The cell is a basic structural and functional unit of life. It must show metabolism, respiration, and response to stimuli. Animal cells usually are contained in a lipid bilayer. They have cytoplasm and nucleus. Nucleus and mitochondria are double bilayer bound. But other organelles like in endoplasmic reticulum, Golgi, and lysosome are single lipid layer bound. Yeast has cell walls made of beta glucan and chitin, and plants have cell walls made of cellulose. And now the nucleus contains DNA, which is in form of condensed form. It's called as chromatin. It contains 2 types of chromatin: heterochromatin and euchromatin. Euchromatin is is about 92%. It's the transcriptionally active light staining part. If you stain DNA with a DNA staining dye, you will see light staining part is euchromatin. Heterochromatin stains darkly. Transcription occurs in the nucleus. Translation in cytoplasm, and dividing cells will have a lower cytoplasm is to nuclear ratio, means it will have a higher nucleus is to cytoplasm ratio. And nucleus contains a darkly stained area called as nucleolus, which is basically the site of ribosome biogenesis and also the site of tRNA, snRNA, and rRNA processing. Nucleolus also contains snORNAs, which modify tRNAs, rRNAs, and snRNAs. And in splicing occurs inside the nucleus. Usually it occurs concurrently with transcription. So as the transcription produces an mRNA, the splicing machinery finds and sits on the mRNA and causes the splicing. It basically mostly occurs by help of spliceosome, which consists of small nuclear RNA and small nuclear ribonucleoproteins. Then this entire machinery, which causes mRNA transcription and associated modification of mRNA, is called as heterogeneous nuclear ribonucleoprotein, or HNRNP. Now, these are important for your short questions, for your MCQs, for exams. And we also find that in some highly dividing cells and transcriptionally active cells like neurons, the nucleus contains dark structures called as causal bodies, which are sites of snRNP, or small nuclear ribonucleoprotein recycling, and causal bodies are also the sites of telomere biogenesis. This is just a basic refresher concept. I'm sure you all have read a part of it. So in order to understand splicing, we first need to understand what is a gene, and let's see how the concept of the gene has evolved. So I'll suggest to the students that to keep your mind open. Biology is a relatively new subject. We have progressed bounds and leaps in the last 10-20 years, but actually biology started maturing from, say, 1970s or 1980s. Physics and maths, chemistry are much mature subjects. They have developed 1880s, in 1900s, they have developed quite a large, as compared to biology. And so what you learn today is is the current knowledge today, and it might evolve, and things what you know today might turn out to be false some days later. So there's nothing wrong in that. Biology is an evolving subject. We are actually, biology is the most current subject now, especially after the pandemic. It is very relevant. So let's see how the concept of gene has evolved, how people have described gene. So it started with Mendel, who described inheritable elements or cell elements, following a set of rules in 1886, and these inheritable elements were given a name by Wilhelm Johansen in 1909 as the word gene. So this is also relevant for the MCQs questions, multiple choice questions, or other competitive exams. Then in 1957, Watson Crick and obviously Rosalind Franklin, we do not attribute much of the credit of structure of DNA to her, but it's actually Watson and Crick relied their entire hypothesis on the plates that were given by Rosalind Franklin on the the X-ray crystallography plates of DNA. So Watson, Crick, and Franklin, they gave the structure of DNA in 1957 in a seminal Nature paper, for which Watson and Crick were awarded the Nobel Prize. And they gave the idea of the central dogma, which is DNA forms RNA, which then it translates into protein. And in 1957, the same year, Seymour Benzer coined the term cistron. This term comes from the cis-trans experiments that were done. And in this, famous lecture series which is called the Symposium on the Chemical Basis of Heredity. So he gave this term. So this is just again for your MCQ purposes. And then we have the function, the definition of the gene. This is taken from Genes 12, the book by Benjamin Levin. The gene is a functional unit of heredity. Each gene is a sequence within the genome that functions by giving rise to a discrete product. It can be an RNA, or it can be further translated to a polypeptide. The importance is that it must be functional. So there are genes that might produce an RNA that does not translate into a protein. Like if you have a bunch of examples, you have small nucleolar RNA, long non-coding RNA, pVRNA, microRNA. So there are the different designated RNAs that are produced by the cell, but they do not translate into a protein. So today the concept of the gene is where it stands. Someday scientists might find some other functions or something important aspects. So things evolve, and this is where our knowledge is currently present. Now, this was what was unknown till 1957, and people are starting to look for genes, for assigning proteins to genes. And when they were looking for genes, they've identified ways of trapping the mRNA and then using this mRNA to hybridize and find the gene. And electron microscopy was an important method that was developed during this time for looking at very small molecules. And some scientists started looking at DNA and RNA using electron microscopy. And of them, Dr. Roberts and Philippe Sharp, these are the two scientists who were independently working. So they were not collaborators. They were independently working, who discovered that genes are split. So this has actually occurred about 20 years later, because in 1957, Watson Crick gave DNA to RNA to protein, and in 1977, about 20 years it took, because the other techniques that were used to look at DNA or do experiments where DNA did not develop at that time in 1957, but by 1977 they had developed so that people could start looking at DNA and RNA at the microscopic level. And for this discovery in 1977 by Robert and Sharp, they shared the Nobel Prize in 1993 for this discovery of split genes. So this is a very important and like a fundamental concept in molecular biology. And in the next year, so 1977, split genes were discovered. In 1978, Dr. Walter Gilbert, another Nobel laureate, he published a beautiful commentary in Nature. It was to describe why genes are split. So it's basically not so much of experimentation, just to give an idea or the concept that why are the eukaryotic genes split. And he basically gave two important concepts. One was that changes in junction of genes of introns and exons due to mutations can lead to origin of new genes. So that's why nature has given this mechanism. It's an evolutionarily important function that the body has. And second, he gave the important concept of alternate splicing, that the same gene can give rise to alternate gene products. So you can understand the importance of this. It's like he just knew that genes are split, and from that he thought over it, and this is just a commentary, and which later were kind of found out to be true by scientists. So this is a very important step in the understanding of the concept of split genes. And then we will look at how split genes were actually discovered. We'll look into the experiments that were done by Sharp and Roberts. So I'm going to show the experiment that was done by Dr. Sharp. He was working on adenoviruses, basically adenovirus 2. He was trying to look at the DNA using electron microscopy. Now, if you look at the structure of adenovirus, it has this major proteins called as hexon and penton. And then there is a fiber-like structure, and then there are minor proteins, and then there are other coat proteins. Now, the major protein, capsid proteins, and hexon and penton, and Sharp could isolate hexon mRNA, and he tried to hybridize this mRNA and try to find or look at the gene that of this hexon gene that is present in adenovirus. And he found that on the right hand side, you can see the picture. This is actually from the actual paper, which was published in 1977, where it shows the electron microscopic picture of the DNA RNA hybridization. So he took the RNA, labeled it, and hybridized it back. So DNA is double-stranded. If you provide conditions, heat it, it will denature. So that means the two strands will break up. Now in that separated or single, it opens up at certain places. It does not open up entirely. It opens up in certain places. And then if you put RNA in the mixture, this RNA, and then you cool the mixture, then RNA will go and bind to the single strand of the DNA to which it is complementary. So the RNA will only bind where it is complementary to the DNA. So wherever it is not bound, the DNA will form a complementary loop, or which is called as the R loops. So this hybrid is actually, if you see the structure here, the red part which I'm drawing is here, is actually this area, and whatever is are these areas are basically the loops that were the large loops that were formed due to the non-hybridization between the mRNA and the denatured DNA. So from this, he understood that this hexon mRNA is actually very small in size compared to the hexon gene. That's why that means that the hexon gene is much larger. It produces an mRNA which is larger, but the final mRNA of the hexon gene does not contain all the region of the hexon mRNA. It does not contain all the region of the hexon gene. So that's why he found that the hexon gene was bigger in size. That means the gene is somehow split into regions that are finally encoding or into the protein, and other regions which do not translate into a protein. And now it is known that around the 36 megabase of DNA, or 36,000 base pairs of DNA, is basically one base pair or one megabase is actually a approximate size of a gene. So ideally it should code around 40 genes, but now it is known that adenovirus DNA can give rise to more than 900 transcripts by alternative splicing. So this is where the fundamental concept of split genes were discovered. Somebody, could you kindly mute the microphone? It's going. Okay, so I'll next we'll just go through the concept of genes. In most higher eukaryotes and some viruses, the genes encoding for RNA products contain regions which are not present in the final mature transcript. So these regions which are intervening, they are present in the gene but do not integrate into the final or mature RNA transcript. These regions are called as introns. The regions surrounding the introns, which are integrated into the final RNA for its function, are called as exons. So they need not code for a protein, they can code for the functionality of the RNA. So introns and exons need not be expressed into protein, they are just defined based upon their presence or absence in the final mRNA. Okay, so having split genes allows eukaryotes to produce a large number of proteins. So in human, if you see, there are only 30,000 genes till now described. There every year you find that this is increasing. It codes for about 90,000 different, so 3 times the amount of proteins as the genes that we have, and about 200,000, so around 6 times of different RNA products. So the most genes are found to have multiple kind of transcripts, and basically giving a definition for exam purposes. Splicing is the post-transcriptional process. So we need to see the importance of the words for your definition purposes. Splicing is the post-transcriptional process of removal of intervening regions of RNA from the premature RNA transcript and joining of intron adjacent regions of RNA called exons, and that appear in the final mature RNA. And this is taken from, I think, one of the books that I was referring, and splicing occurs in the nucleus of the RNA. So like we were told when I was in college, I think location, structure, function, that's how you should define any definition of any concept, like any organism or any function that is occurring in a body. So if it should say where it occurs, so the nucleus and what function it does and how it does. So location, structure, and function all is defined in the concept. So splicing can be spliceosome mediated, or it can also occur by auto-splicing or self-splicing in certain kind of RNAs in eukaryotes, and these are called as catalytic RNAs or ribozymes. We will look at those sometime later. And important concept is splicing is a way of gene regulation, because splicing can alter RNA's location, post-translation and modification, its stability, and as well as its function. So some facts about split genes in eukaryotes is just for your knowledge. It's mRNA is going to have between 0 to 362 introns. Transfer RNA genes may have either 0 to 1 intron. And there are a large number of intron-less genes, and this is the distribution of the intron-less genes in the human genome that is shown. You can see even Y chromosome, which is a very small chromosome, still has some intron-less genes. And there are about 4,000 intron-less genes in human. And this is another interesting thing, that this is till date known the gene that has the largest number of splice variants is the Drosophila DSCAM gene, or Down syndrome cell adhesion molecule. One single gene can actually produce 38,000 different variants of splice. So this is the largest number of splice variants that is known till date for one gene, and this is in Drosophila. And it's actually, it shows the basic calculation is shown here, how many introns and how many exons, and they can recombine within themselves, and this is reported that it can produce about 30,000 different genes. So looking at specificity of splicing, splicing, we have two parts of splicing, intron and exon. You remove the intron, you join the exon, and so there's the importance of the intron-exon boundary in splicing. Now most mRNA have a GUAG sequence, the GU on the 5' splice site and the AG sequence on the 3' splice site, and this phenomena of the presence of large number of these GUAG sequence was discovered by Chambon in 1978. I don't know how to pronounce it. Could be Chambon in French. I don't know what's the exact pronunciation, but this is called as the GUAG rule, and this site is called as and this follows for most of the mRNAs or the RNAs that we see in eukaryotes. But there are also a small number of introns which follow an alternate, which is the AUAC intron. So there's a different kind of intron that follows a different rule. And now this is also like not universal. I mean, you might find other introns that do not follow this rule, but it is generally the rule that maximum number will be GUAG introns, a small number will be AUAC introns. And for the sequencing has revealed that this set interjecting intron sequence, usually there is a branch site. So this is a sequence which is called as the branch site. It has a conserved adenosine residue which serves as a hydroxyl moiety donor and starts the splicing process. And you can see that this branch site sequence is quite conserved in yeast. It's UAC, and in higher eukaryotes it can vary a bit. It can be Y, which is Y where Y is a pyrimidine, and then YN denotes a string about 9 pyrimidines, and R is another in purine. So there is consensus sequence around what it can be or what it should be in eukaryotes. But usually the important thing to remember is that there is a conserved sequence which we know very sure is conserved in yeast. In eukaryotes it varies, and there is a conserved adenosine residue in most RNAs that serves as the branch point donor, branch site where splicing occurs. So now we will look at how splicing occurs. Splicing usually occurs via lariat formation and true transesterification reaction. So now if you look at the intron and exon, so this is the intron, and here you have the branch site in which there is a conserved adenosine. There's a 5' GU sequence, which is the 5' splice site, and an AG sequence on the 3', which is the 3' splice site. So GUAG following Chambon's rule, and there is a consensus sequence in higher eukaryotes, which can be pyrimidine, any nucleotide, pyrimidine, purine, and adenosine conserved, and then pyrimidine. And these percentages show the probable percentages of presence of purine, of pyrimidine or purine in those respective residues. So this sequence can be a pyrimidine 20% of the purine at 20% of the time. So this is basically, but you will see that this adenosine important residue is very conserved. If you mutate this residue, splicing will not happen, or it might start somewhere else downstream. So this adenosine residue serves is called as the conserved branch site, and this is less. I mean, this is where the starting of the splicing reaction starts. So splicing starts when the 2' hydroxyl moiety of the conserved adenosine starts a nucleophilic attack on the 5' guanosine. So this conserved moiety starts a nucleophilic attack on the 5' guanosine, and then a first transesterification reaction occurs. It's an SN2 type transesterification, which means it's a substitution and bimolecular and nucleophilic attack. And this causes, and if you have read in chemistry, SN2 causes a movement around the chiral center of the carbon, so that this actually twists the RNA so that the 5' and the 3' sites come closer for the next reaction. So so once this hydroxyl ion does a nucleophilic attack on the 5' splice site, it gets joined to the guanosine on the 5' and forms a structure like this. And this structure here is joined by a 2', 5', 3' phosphodiester bond, and then this brings this 5' closer to the 3' side. This is because there is a rotation of axis around along the carbon on the adenosine residue. And then the second transesterification reaction occurs where the 3' hydroxyl that is remaining on the 5' 3' hydroxyl moiety that is remaining on the 5' end of the DNA now attacks the 3' phosphate on the 3' splice site. So this moiety attacks the phosphate on this side, and then the second SN2 transesterification reaction occurs, and the two exons are ligated. It is important to note that this phosphate, which actually belonged to the intron, is retained by the exon, and these two exons are ligated, and the intron is released as a lariat, which completes the splicing process. Now, this is basically how splicing occurs between one intron and two exons, and how these two exons are ligated. And this occurs along the stretch of the mRNA or the RNA in eukaryotes. Now we'll look at what are the different types of introns. Basically, we know that transesterification introns are the maximum. Most introns follow this transesterification process, but there are certain exceptions. There are non-transesterification introns. So basically, you can split into two types: transesterification, non-transesterification. Within the transesterification, there are most follow spliceosome dependent, and there are others which are spliceosome independent or self splicing or ribozymes. Now within the spliceosome independent, there are two kinds, which uses the major spliceosome, which uses the which is the major kind of spliceosome that is present in human. We'll look into that a little later. And there are the minor spliceosome or U12 dependent introns. And then within the ribozymes, you have class 1, class 2, class 3, and twintrons. And to look at, you will find this reference very well described in Twyman in Advanced Molecular Biology. Basically, instead of adenine residue, the group 1 uses a guanosine residue, which is present here, and that acts as the splice site donor, and there is no lariat formation. So you can see this is what basically the reaction occurs in group 1. And then you have the group 2 or class 2, where this it occurs similar to what we see in spliceosome mediated, but there is no spliceosome involved. Rather, this occurs on its own by autocatalysis, because the RNA itself contains catalytic properties, and this occurs via lariat formation. Now, each of these two, the size of the intron is around 200 to 250 base pairs. Now, this was known till 1998 or 2000 that this is basically the two types of introns. And then some scientists discovered that no, there is another type of intron, which is a self splicing class 3. The only difference is that it's smaller. So it was again described as a new class, because you would expect that in order for the proteins to sit on and recognize the intron and exon, they need to be of a certain size. So 200 base pair was considered as the required size that the proteins would need to identify this intron boundaries and splice it. But then there was another small class of introns which are identified as class 3. They occur in the same mechanism as class 2, only that the intron size is smaller. It's 100 to 120 base pairs. And then there are non-transesterification introns, which are eukaryotic rRNA, tRNA introns. And just a small note that if you mutate this branch site in type 2 splicing autocatalytic introns, what happens is that this reaction starts without the presence of this adenosine, but by hydrolysis. So hydroxyl ions serve as the nucleophilic attack. So water basically provides the hydroxyl moiety that starts the nucleophilic attack. And this was also seen in vivo splicing in 1998. There's a paper published in Nature which shows that in certain introns in group 2, where the splice size is mutated, you can see the transesterification reaction starts via hydrolysis, and there is no lariat formation there. And this is basically the type of introns. It's needed for your long questions, or but the very good reference is Twyman, and I think that book has the best reference for these different type of introns. And now let us look at the major class of splicing which occurs via spliceosome. The spliceosome is a large machinery. We will look into the structure of it and what it's composed of, and the two kinds of spliceosome that exist in eukaryotes. So Brody and Abelson in 1985 discovered that the lariat-shaped intermediates in yeast, so they were studying yeast splicing. They found that the lariat-shaped intermediate which was produced during splicing is always accompanied by a heavy protein mass which sedimented at 60S, and this is called as the spliceosome. And in the same year, Sharp, who discovered split genes, he isolated spliceosome for human cells, and in yeast they were found out to be 40S particles. In human it is 60S particle. Now the spliceosome is a very large machinery. It's 12 megadalton. If I compare any human protein, it is usually in the range of, I would say, 50, 60, 100 kilodalton. So you can see it's 10 times as large as actually 120 times as large as a human protein. You can see that spliceosome is actually, it's a fluid concept. It's the structure that's called as a spliceosome, but the composition changes over time. So it's a ribonucleoprotein complex that is composed of 5 standard mRNAs, U1, U2, U4, U5, and U6. There is a U3 RNA, but that's not part of this spliceosome. You'll find a slide where I have shared what is U3 snRNA, and these are composed. So the major spliceosome is composed of 5 snRNAs and 41 snRNA associated proteins called as snRNPs, and these proteins are called as SM proteins. The reason is that they are identified by a class of antibody called as SM antibodies that were identified in autoimmune disorders. Now, along with this, there are 70 other splicing factors and 30 other helper proteins called as SR proteins that get associated as a part of this complex. So it's a huge complex which contains 5 snRNAs, 41 snRNA associated proteins or snRNPs, 70 splicing factors, and 30 other proteins. So hundreds other proteins, 40 proteins here, so 140 proteins and 5 snRNA. That's basically, you can see 145 proteins and 5 snRNA. This huge complex is called as spliceosome. And in eukaryotes, the major spliceosome is called as the U2 dependent spliceosome because it contains this U2 snRNA and its associated proteins, and 99.5% introns are actually spliced by this snRNA, this spliceosome, and mutations of this major spliceosome have been found in a lot of cancers, example chronic myelomonocytic leukemia, CMML. And there is another class of spliceosome which is called as a minor spliceosome, where you can see that the U1, U2 are not there, but this is U11, U12, U4, U5, and U6 ATAC snRNA, and this is called as U12 dependent spliceosome because this contains U12 instead of U2, and this splices around 0.5% of introns in humans and eukaryotes. And mutations of this spliceosome have been found to be associated with a lot of disorders. One of them is called Tavy-Linda syndrome or TALS. This is just for your knowledge or example, short question or MCQ purposes. And let us now look at how the this is just a video to show that the spliceosome has an uncanny similarity with the ribosome. Both are snRNPs or contain RNAs as well as proteins. And if you look at the structure, you will see that it will seem that they have evolved from similar kind of genes, because you will see that they have a central core where through which the RNA kind of slides in, and then they do their work on the RNA. And this will have a video from the original video that I had taken.
Consider the spliceosome, the cellular machinery which carries out splicing, versus the ribosome which synthesizes protein, built from RNA.
So you can see both have a similar structure. If you see, they have this 24 nanometer size, around 30 nanometers. So around 25 nanometer by 30 nanometer and 20 nanometer deep. They have the structure, looks kind of similar with a catalytic site, a kind of like a donut shaped structure in the center through which the RNA slides, and they perform their function. So you can see the beauty of evolution. You will see that ribosomes evolved earlier, and then splicing doesn't occur so much in prokaryotes. There are examples, but it's not the predominant structure. But you can see as evolution has occurred, ribosome-like structures which work on the RNA has evolved along with the presence of split genes. So you can just to show a structure between the similarity and to show the beauty of evolution, that how proteins that do similar work look similar and have similar dimensions because they do the similar kind of structure, similar kind of function. So now let's look at how the splicing, spliceosome mediated splicing occurs. So this is a cyclical pattern. It starts with an assembly, disassembly, and recycling of the components. So as you know, cells are very intelligent. They don't like to waste energy. So any protein that is utilized in one function or one reaction will go back to the repository where it is present and come back at another time and do the work until it is degraded. So every protein has a lifetime. The cell does not always produce protein. It keeps protein and makes a stock of the protein. The protein does a function multiple times, and then it degrades, or it might get degraded for after one time. So these components of the nucleosome of the spliceosome are also recycled. And it's let's see how the spliceosome occurs. I think this is the most important part to understand. So there are 5. This is for the major spliceosome. So we must remember there are 5 different snRNPs and their associated proteins, and this is called snRNAs and their associated proteins, which are snRNPs. So there are U1 snRNP, U2 snRNP, U4, U5, and U6 snRNP. And these snRNPs, the snRNAs that are composed, they have complementarity with the 5' splice site or the branch site or the 3' splice site, which allows them to bind to the splice site and then do their function. So the first step is the binding of the U1 snRNA at the 5' splice site, and the U1 snRNA interacts with its sequence to the because it's complementary to the 5' splice site. It binds to the 5' splice site of the mRNA, and a stretch of 4 to 6 bases of this sequence is homologous, is complementary, and it binds there. And then the U1 snRNP binding is stabilized by binding of 2 other proteins, which are called branch point binding protein, BBP, and MUD2, which is basically, these are nomenclatures in yeast because that's where splicing has been most studied. And then this binds to this branch site, which stabilizes this complex, and this function is not ATP dependent. This is called as the E complex. So the presence of the U1 snRNP on the 5' branch site and the BBP and the MUD2 on the branching site is called as is a stable complex which is not ATP dependent. So if you mutate this branch site, the branch point mutant will there will be no initiation of splicing. So you can either block splicing by mutating the branch point, or you don't, because you don't need ATP. The next step starts with need of ATP. So the next step is the binding of the U2 snRNA, which binds on the splice site. So the splice site already has BBP and MUD2 bound to it, and then it allows the binding of the U2 snRNP, which is again complementary to the branch site, and this requires ATP hydrolysis. So if you block ATP here, this U2 snRNP will not bind, and this complex is called as the A complex, and this is also called as the pre-B complex, because I mean, different scientists have named it differently, but we'll usually follow one nomenclature. Like the U1 and BBP is E complex, followed by binding of U2, which is the A complex or pre-B complex. The next step, the primer of U6, U5, and U4 bind to this structure where already U1 and U2 snRNPs are bound. Now you have to, so if you see the structure of the U4 and U6, these are actually a di-snRNA. So U4 and U6 are bound together, and to this complex, the U5 binds, and this forms like a trimeric complex. It's a heterotrimer, and once this binds, so that means in order to for the U6 to bind, the U4 has to leave the U6, and U6 will go and bind to the mRNA, because U6 is most important snRNA in this complex, and this step requires ATP hydrolysis. And so the B1 complex is formed when the trimer of U5 and U4, U6 snRNP. So U4 and U6 are called as di-snRNP because they bind kind of together. They are always together. They go and bind to this A complex, and this is called as the B1 complex. And this complex is actually regarded as the spliceosome because now it contains all the component snRNPs that is required for the assembly, and this is the activation stage of the spliceosome. Now splicing will occur. So the binding of the U6 snRNP detaches the U1 snRNA from the 5' splice site, and also because U4 and U6 are bound together, once U6 goes and binds to the RNA, the U4 is also released. So U6 binding detaches U1, and because U6 binds to the RNA, U4 is released, because initially U4 and U6 are bound together. And this converts the complex from B1 to a B2 complex. And this is where the magic happens, because when the U6 bind, it twists the RNA and brings the 5' and the 3' region closer. And then this occurs in the presence of ATP, and then the 2 transesterification reaction occurs. Now, this transesterification reaction, the first part when it occurs is called as the C1 complex. The second transesterification, when the joining of the 2 exons occur, is called as the C2 complex. And now this is catalyzed by the U2 U6 snRNP complex, which is still bound to the mRNA along with U5, and it is dependent on ATP as well as the presence of magnesium ions. In the next step, the exons are already joined, the lariat is formed, and the lariat leaves bound with U6, U2, and U5, and the 2 exons are released. That's how basically the splicing occurs. And these U2, U6, and U5 are released. They go back to the pool of proteins of the spliceosome to complete the next cycle, and the lariat of the intron is released, and it gets degraded. And the cell reuses the nucleotides that were present in the intron. So you can see the cell utilizes all the components for another round of cycling. And this BBP protein, you can see in the footnotes, BBP is also called SF1 in eukaryotes, and in higher eukaryotes, the function of MUD2 is replaced by U2AF. I mean, I have shared the slides with Dr. Bandyopadhyay. You can get the slides from him. And so this is how basically the assembly and disassembly and recycling of the spliceosome occurs. The next step is just to show a video of how this recycling occurs. There's also audio from the original video. Oh, okay, there's no audio. So the first is binding of U1, followed by binding of U2, formation of complex A, and then U4, U5, and U6 bind together. Then U1 and U4 is released, forming the complex B, which is kind of the active spliceosome, and then it starts the first catalytic transesterification, followed by the second catalytic transesterification, and then the intron is released and as a lariat, and then all the ribosomal, all the snRNPs are released and recycled. So this is how splicing occurs. Now let's see how the splicing start. How do the spliceosome machinery know where to sit? So they can either start by recognizing introns or by recognizing exons. Now where it can start either way, but in introns which are less than 250 nucleotides, the spliceosome machinery goes and binds, and because they are large structures, the U1 and U2 interact and they recognize the two boundaries of the intron. That this is called as intron definition, where the sizes are larger, then the U1 of one which is bound to the intron exon boundary recognizes the other U2 which is bound to the other end of the intron by looping around, and this is called as exon definition. Basically, here the spliceosome machinery is identifying the boundaries of the intron, and here they're bound to the exon and identifying the boundaries of the exon. So this will interact with the protein here, and this is called as intron, either intron definition or exon definition, depends upon the size of the intron. And now I'll go to the I think this is the last but one slide, which is the alternative splicing. Now alternative splicing is a beautiful phenomena because it allows the cell to conserve energy. Now cells are very conserved, they conserve and try to conserve energy at every step. They are very finicky about expenditure of energy. So if you have, say, if you want to produce 50,000 different proteins, and a cell has one gene for every protein, you need to have 50,000 genes. When this cell duplicates, you need to use a huge amount of nucleotides to replicate the DNA. So that's why cells have evolved genes which can have alternative splicing. So in order to produce 50,000, you need to have, say, around 10,000 different genes. So it's an evolutionary way of conserving energy, and this concept has evolved, and cells have used it to their maximization of utilization of proper utilization of energy. Now, alternative splicing was identified in 1980s in the mouse immunoglobulin M heavy chain gene. So as you know, there is one class of immunoglobulin which is called as immunoglobulin M or IgM. This IgM heavy chain exists in two forms, a secreted form and a membrane-bound form. Now the secreted form is shorter and the membrane-bound form is a little longer. The membrane-bound form has an extra residue on the carboxy terminus where it is hydrophobic and it binds to the membrane. Now, using hybridization, Hood found that these two proteins are encoded by two separate mature RNAs, which are actually identical at the 5' end. So this 5' end is identical in the two RNAs, only they differ in the 3' end. Then he came to know that, yeah, this is an example of alternative splicing, which was actually if you see in 1978, 2 years before this was identified, it was already described by Dr. Walter, I forgot the name. So this was described 2 years after the concept was given, and this it was described in a mouse immunoglobulin M gene. And so the process, what is alternative splicing? This process by which a single pre-mRNA can get spliced into two or more alternative forms of final mRNA by including or excluding specific exons in the different forms of the mature mRNA is called as alternative splicing. Alternative splicing occurs in most eukaryotic genes. 90% is like an average number, but if you look at humans, 95% of genes are alternatively spliced. And this is basically a method of control of splicing. Now, splicing can be regulated by different proteins. They are called repressors and activators. This is just for your knowledge. I don't think this is necessary at this stage, but there are repressors and activators which will either allow a certain splice site to be shown to the spliceosome, or they will repress. They will go and bind to a splice site and not allow the spliceosome to go and bind there and splice. So a different splice site will be chosen. So the mRNA will become longer than so. There are different proteins called as activators and repressors. There are also enhancers called as exonic splicing enhancers, or ESCs. There are different regulatory elements within the DNA that allow or control splicing, and this is called as the splicing code that basically governs how splicing will occur under certain cellular conditions. We'll see an example in later. And splicing is also a method which is a process which is interrupted by transposons. As you know, transposons are jumping DNA. They jump from one DNA to another, and there are also other proteins, RNAs called as pRNA, pRNA or PV interacting RNA. And there are also different kinds of alternative splicing. You can look up retrohoming, and this is present on the slide, but I'm not going to present it. You can read it up later. That where the intron gets, so retrohoming is a process where the intron gets reintegrated into the DNA by reverse splicing. So there are different alternative forms of splicing that have been described. And to summarize, so splicing is a post-transcriptional process of removal of intervening regions of RNA, which are called introns, from the final premature RNA transcript, and joining of intron adjacent regions called exons that appear in the final mature RNA. And splicing occurs in the nucleus. Splicing can be spliceosome mediated or auto splicing in certain kinds of RNAs, which are called as catalytic RNA ribozymes. Splicing requires the 5' and 3' splice site and a branch site, and splicing starts by the nucleophilic attack of 2' hydroxyl residue on the conserved adenosine residue on the 5' branch site, resulting in first transesterification and lariat formation. The free hydroxyl group on the 5' splice site now attacks the 3' phosphate on the 3' branch site, causing the second transesterification, where exons are joined and introns is released as a lariat. So this is how the splicing occurs by two processes of two transesterification reaction and lariat formation. And spliceosome is a large 12 megadalton complex composed of snRNAs, SM proteins, and SR proteins, and other splicing factors. 5 snRNAs are present in the major class of spliceosome that we see, U1, U2, U4, U6, and U5, and they have their associated snRNPs. snRNAs are complementary to the splice site, which allows them to bind along with the protein and execute the splicing. Splicing starts by binding of U1 snRNP to the 5' splice site, followed by binding of branch site binding protein to the branch site, which has the conserved adenosine residue. Then the U2 snRNP binds to the branch site, and next U4, U6, and U5 bind to this complex. The U6 binding releases U1 from the 5' splice site, and the U4 is also released because U4 and U6 are initially bound together. So as U6 binds, the U4 is released, and U6 binding causes U1 to get released, and this causes a reaction which causes the two transesterification reactions, and the exons are ligated, and the intron forms a lariat, which is left with the U2, U5, and U6, which are then recycled. The intron is degraded, and splicing requires the presence of ATP and magnesium, except for the first place where the U1 and U2 are binding, the formation of the E complex or the early complex, that is ATP independent. And mutation of the splice site will obviously reduce frequency of splicing, or an alternate splice site will be taken up. And splicing can occur by either intron definition or exon definition. Introns can be spliceosome dependent or self-splicing. Spliceosome dependent introns can be U2 dependent or U12 dependent. Self-splicing introns or ribozymes can be class 1, class 2, or class 3, or twintrons. And finally, the process by which a single pre-mRNA can get spliced into 2 or more alternative forms of final mRNA by including or excluding specific exons in the different form of mature mRNA is called an alternative splicing, which occurs in 90% of eukaryotes, around close to 95% in human So this is basically what I had. I have more. Shyam, do we have time for more?
I think we have run out of time.
We have run out of time? Okay. Okay, so this is, sorry, I took up more than that, and this is where I will.
No, that's perfect. That's perfect. You are good.
Okay, so this is where I had. I wanted to present, and okay, so it's 7:30. So yeah, this is what I had to present. I hope it's helpful, and if you have any questions, I'll be happy to take them.
ডক্টর সৃজিত খান মহাশয়কে তার মূল্যবান তথ্য নির্ভর ও মনোজ্ঞ বক্তব্যের জন্য অসংখ্য অভিনন্দন জানাই। এখন ছাত্রছাত্রীদের কোনো প্রশ্ন বা জিজ্ঞাসা থাকলে তোমরা বক্তার সঙ্গে আলোচনা করতে পারো। ছাত্রছাত্রীদের কোনো প্রশ্ন থাকলে বা জানার কিছু থাকলে তোমরা বক্তার সঙ্গে আলোচনা করতে পারো। আমাদের হাতে অল্প একটু আলোচনার সময় রয়েছে, তোমরা আলোচনা করতে পারো। হ্যালো, কারোর যদি কোনো প্রশ্ন থাকে বা জানার কিছু থাকে তোমরা বক্তার সঙ্গে আলোচনা করতে পারো।
আমি স্লাইডগুলোর আগে একটা পিডিএফ ভার্সন শ্যামকে পাঠিয়েছিলাম, তো ছাত্রছাত্রীদের সাথে শেয়ার করেছে। যদি আরো কোনো প্রশ্ন থাকে যেগুলো হয়তো তোমরা পড়ার পর মাথায় আসবে, সেগুলো তোমরা মানে আমাকে ইমেলও করতে পারো, বা এখন যদি কিছু জিজ্ঞেস করার থাকে জিজ্ঞেস করতে পারো। এই যে প্রেজেন্টেশনটা বানালাম, এটা ছাড়াও কিছু এক্সেস স্লাইড আছে যেগুলো আমি অ্যাড করিনি ইন ইন্টারেস্ট অফ টাইম, বাট শ্যামকে আমি একটা পিডিএফ ভার্সন পাঠিয়েছি। যদি তোমাদের ইন্টারেস্ট হয় তুমি লার্জার ফরম্যাটটা তোমরা চেয়ে নিতে পারো। আর that's it.
Can I ask one question?
Yeah.
I think so. The organisms which do not have introns and the organisms which do have introns, both of them are evolving. So
yes.
What is your comment on that?
No, I think splicing is actually efficient for the cell. So organisms that do not have introns, I mean, will continue to not have introns. They might acquire the property of having introns, introns in subsequent evolutionary stages. I think I see splicing as a beneficial. So I mean, there are reports where bacteria have, which are not supposed to have introns, and also in some plants which are slowly being discovered that they have introns. Earlier they were reported to not have introns, but they have introns. And actually, if you see the slide deck that I have sent, there is actually introns can also code for proteins. There is something called as retrohoming, where initially something which was thought to not encode a protein, but you'll see that the intron itself encodes a protein which helps in integrating the intron into DNA. So this is called as retrohoming. You can read more on it on the slides.
আর কারোর কোনো প্রশ্ন আছে? কারোর কোনো প্রশ্ন থাকলে।
I have one query. Thank you, Dr. Srijit Khan, for your valuable speech. We taught that the histone genes are intronless. Is there any reason for this?
Yeah, so I also have, I think it occurs, histone mRNA splicing, I think occurs in the nucleolus. A part of it, processing occurs, but I don't have too much information on that. But it could be that it is, the reason is because they need to be translated faster. So maybe the transcription, because if you see transcription followed by splicing leading to translation takes up some time, and actually they occur concurrently. So as a cell is producing mRNA, it is getting spliced. So at the same time, so as soon as the mRNA starts coming out of the RNA pol 2, the RNA pol 2 has a long C-terminal domain where the splicing machinery is just sitting there and waiting to trap the mRNA inside and then start the splicing. So maybe this takes up some time, and the only way that I can see as the reason why histones have less introns, because if the splicing does not occur, the translation can occur immediately after transcription. And actually splicing also provides a way of identifying whether a mRNA is properly spliced or like has any aberrant mutations or not. So if splicing is not there, there's no chance of any check on the amount of mRNA that is getting produced or translated. But I don't think that's a very perfect answer that I gave. I think, I mean, a better would be to, just for the students would be to just look up in the internet why I think, but this is what I think.
Thank you.
আর কার কোনো প্রশ্ন আছে? আমার মনে হয় যে ছাত্রছাত্রীরা হয়তো এই মেটেরিয়ালটাকে পড়ার পরে কিছু প্রশ্ন পাবে, সেক্ষেত্রে তোমাদের ইমেল আইডি দেওয়া আছে বক্তার, সেখানে তার সঙ্গে যোগাযোগ করে নিতে পারো ভবিষ্যতে। তো আমরা অনুষ্ঠানের একবারে অন্তিম লগ্নে এসে উপস্থিত হয়েছি। এখন আমি বর্ধমান বিশ্ববিদ্যালয়ের প্রাণীবিদ্যা বিভাগের অধ্যাপক ডক্টর নীলাদ্রি হাজরা মহাশয়কে ধন্যবাদ জ্ঞাপন এবং অনুষ্ঠানের আনুষ্ঠানিক সমাপ্তি ঘোষণার জন্য আহ্বান জানাচ্ছি। নীলাদ্রি।
Thank you, Dr. Mojumdar. Good evening to all. Honorable members of the executive body of the Zoological Association of Bardhaman, Professor Subroto Roy, Dr. Subroto Mondol, Dr. Dilip Guho, Dr. Manavesh Mojumdar, Dr. Saroj Kumar Ghosh, and Dr. Samdas Bandyopadhyay. Our resource person, our chief guest, Dr. Srijit Khan, beloved students and other dignitaries. It is a customary to conclude the session with a vote of thanks. First of all, I would like to convey my sincere thanks to the executive members of the Zoological Association of Bardhaman for approving this online lecture series. It's a good endeavor from the association for the societal benefit at large. I am thankful to Professor Subroto Roy, President of the Association, and Dr. Subroto Mondol, Secretary of the Association, for their continuous inspiration and effort to make it successful. Special thank goes to Dr. Manavesh Mojumdar and Dr. Saroj Kumar Ghosh of Bijoy Narayan Mahavidyalaya, Itachona, Hugli, and Dr. Samdas Bandyopadhyay, Vivekananda Mahavidyalaya, Bardhaman, for arranging this online lecture series for the students of different undergraduate semesters for a period of more than 2 months. Actually, they are the main architect of this kind of activity. We are grateful to all our beloved students. Without their active involvement, participation and enrollment in this lecture series, the program could never be successful. Hopefully, they will make it successful. I convey my sincere thanks to Dr. Srijit Khan, a resource person, for his valuable speech, and hopefully students will be able to understand the split gene concept. And finally, I convey my thanks to Google Meet platform, using which the online lecture series will be conducted in near future also. With these few words, I conclude the session.
ধন্যবাদ নীলাদ্রি। শুধু একটাই কথা, শ্যামদাস বন্দ্যোপাধ্যায় বর্ধমান রাজ কলেজের অধ্যাপক।
সরি।
হ্যাঁ, রাজ কলেজের অধ্যাপক। আমরা তাহলে অনুষ্ঠানটা এখানেই শেষ করছি। সবাইকে অসংখ্য ধন্যবাদ।
তাহলে কি আমরা